pronounce, actually. You did. So you did great.
Perfect, wonderful. So we will start with you, Michela.
Wonderful. I'll start sharing my screen. Okay. Okay, here we are. Thank you very much for your invitation and for all your great work. It's great to see your conference happening every year. So today I will talk about the project ProDGNE, and I will be accompanied by Fabrizio Pertusati, which I'm very glad that you are going to meet today. And my presentation will be followed by his, who will explain more about the prodrugs and why we are working on this. So in my introduction, I'm going to talk about the role of patients in this project and why it is essential to get to a solution for GNE myopathy and in general for all rare diseases. So the importance of patient involvement. Okay, so how can we accelerate research in GNE myopathy? This is the question that I asked myself some years ago after being diagnosed with GNE myopathy. And it is something that I applied in this project to make it happen. So in the world of rare diseases, we have two options. The first option is waiting for research to make its course, which of course is not an option because we need a therapy and a treatment as soon as possible. And if we wait for research to make its course, it means that we want to make research without the patients, which is not an option because at the central partner in research should be the patient, because eventually is the one who needs the treatment and who is living with the disease every day. So when I thought about this project, my idea was, of course, to make it from the start with patients, making sure the patients take action as partner and are full decision makers in research. So in research, it's very important to understand that all stakeholders need to be involved, and patients are the primary stakeholder to be involved. And only this way we can get to a treatment as soon as possible without failures. And I will explain you more about this and why we think that involving the patient in early research is essential to avoid possible problems along the way of research. So how can we involve the patients and what is at the base of this project? What I wanted to understand in order to help researchers were the challenges in developing therapies for GNE myopathy, which I think are essential to be understood by patients as well. So I want to go through this slide very quickly because I don't want to take too much time, but the situation in GNE myopathy is quite challenging as it is for many rare diseases. So we don't have, we have limited animal models, we lack reliable biomarkers, which make, of course, clinical research very, very difficult. We have limited knowledge of natural history, so it is important that patients understand that research can be successful only if we understand the disease very well, and it's not really easy when we have so limited number of patients worldwide. Our disease progression is low, so we noticed this in the Ultragenyx trial. Designing clinical trials is very difficult because it's very difficult to determine treatment effect since the progression is so slow. We don't know much about the mechanism of the disease. This still poses many challenges in all researchers working in GNE myopathy. And then we know GNE has a role in sialic acid biosynthesis and regulation, but we don't know what other roles there are in cellular function. So this is to explain you how it's so complicated to develop treatments for GNE myopathy. So what questions did we want to target in GNE myopathy? The first question that I asked myself and that I posed to researcher is okay, the Ultragenics trial failed. What did we see in there? Was there a lack of therapeutic effect, or the protocol design was very well developed? So something that we need to look into, and that has been addressed now with the MANAC trial, and we hope to have positive results. Another question, can we explore therapeutic strategies that stays within the substrate replacement therapies options? So is a substrate replacement option still a solution to be explored for GNE myopathy? What did we learn from the Ultragenics trial? So this is exactly what I wanted to address in this project, and exactly we want to address absorption issues encountered in these clinical trials. So what can patients do? How can they contribute? Definitely they can contribute as early as possible, and it's very important that they participate regularly in natural history studies to make sure that we don't get to clinical development without having not much data about the progression of this disease. So we got into the European Joint Program on Rare Disease call on developing effective therapies for rare diseases, and this is how everything happened. So after the Ultragenics failure, my objective was absolutely not to stop and to learn from what we learned in this trial and continue, making sure that the efforts in the last 15 years weren't useless, because they are not useless. It wasn't a failure. We learned a lot, and we need to continue our efforts. So in 2019, I saw this article, and this is how I approached Cardiff universities and how ProDGNE started. So improving a delivery for MANAC and making sure that this is improved, but Fabrizio will explain you more. And this is where we are since 2019. We were founded in 2020, and this is all of us working toward the GNE myopathy in ProDGNE. So the focus of ProDGNE, just quickly, and then Fabrizio will focus on the prodrug, is of course to develop an improved substrate approach for GNE myopathy. We want to improve drug absorption and therapeutic effect to address the gastrointestinal adverse events encountered in clinical trials. We want to identify biomarkers through proteomics and metabolomics, and we want to determine, of course, the safest and most promising compound for GNE myopathy. At the same time, we are working on modeling GNE myopathy in zebrafish model. So just to finish my presentation and my introduction, this is the team working in GNE myopathy. So we have Equilibristi HIBM. We have the institutes in Ottawa with Dr. Hans Lochmuller and his team. Then we have Fabrizio Pertusati from Cardiff University, Paula Videira at Nova University in Portugal. Then Dr. Rudiger Osporte in Germany, and then in Italy, Pierluigi Caboni. So this is ProDGNE, and now we will focus with Fabrizio more about the reasons for the prodrug and how our work is going for the prodrug. So you can contact me anytime, and you can find us on Twitter for up-to-date information about our work and what we are doing. So I will leave the floor to you, Fabrizio. I hope I didn't take too much time.
Thank you very much, Michela, for this wonderful and insightful presentation. Now we will go ahead with Dr. Fabrizio Pertusati. He is a lecturer at the School of Pharmacy and Pharmaceutical Sciences at Cardiff University. Dr. Fabrizio, the floor is yours. You are on mute.
I discovered that. Thank you very much. So I was saying, it's a really great pleasure to be invited to this conference. As I really look forward to talk about our science and hopefully to make an impact on GNE myopathy patient. So to take over from Michela, everything was started. Here we go. When we published this paper, and Michela got in touch with us. And Michela is from Sardinia, and my wife over here is from Sardinia. So we had the Italian connection, as we can say. And this also facilitated our connection because in this picture here, we were meeting in Cagliari University, in one of the members of the consortium. And here we, thanks to Michela input and thought, we started to, to build this consortium to take forward this our project that started way back, probably 2 years prior to 2019 as a date of this paper. So before going forward, I'll keep my presentation very simple. I apologize for the scientists in the audience, but just to give you a very broad overview to everyone, which is the rationale behind our approach. So we are talking about drug delivery in the slide here. There are several different methods to get the drug into the human body. But today we focus on oral administration because Mannac is a drug that is taken by mouth. So oral administration of drug is basically the broad topic of this talk. So before introduce this drug delivery, I want to just to set the scene for this talk and just put everything in concept. So drugs are molecules, okay? And we can distinguish in two different kinds of molecules, polar molecule and non-polar molecule. So what is a polar molecule? We have an example in our daily life, sugar cubes, and Mannac, I just pick up this example because Mannac is a sugar, and you can do the experiment at home. As soon as you try to dissolve the sugar in water, it dissolves immediately. Really, really straightforward. If you take another experiment, if you try to take the same sugar cubes and try to dissolve in the bowl of oil, what happens? The sugar doesn't dissolve. Okay, it's a very simple experiment that everyone can try at home. But what are the implications of this factor in drug delivery? So basically, the polar molecules need to be dissolved in water first, but then you need to be able to dissolve in water to reach the cellular target or to be absorbed easily. So if you have a molecule like Mannac, you need to pass through something that looks like an oil bowl, then you can immediately spot that we have a problem. Okay, and this is the kind of one of the two problems that we try to solve with our prodrug approach. So what happened when we take a polar drug like Mannac over here? Remember, polar drugs means it's very likely to be soluble in water, like the sugar cube I just talked about before. This is because we have this group here, OH, okay, they are free, and this make this compound here very similar to water. So similar want to dissolve similar, so this compound here want to stay in water. So when we take the pill by mouth, what happens? This goes through the stomach, okay? There's no problem here because the stomach is basically water. Then it moves to the intestine, and here we start to have this kind of problem. Why? Because the drug at this point face three different challenges. It needs to be absorbed by the gut, and in order to do so, it needs to be fat soluble, okay? Then it needs to be solubilized in the bloodstream, which again is water-based, but the human body has a system to solubilize these fat molecules into the bloodstream. And then, importantly here, you have to cross the cell membrane. In order to do so very easily, you need to have a very, very lipophilic molecule, like resembling an oil, okay? The oil bowl that we saw before. So here is just a very quick representation of what happened. The guts are here. So basically you can see mucus bilayer. Okay, so the polar drug need to cross this part here, and then to reach basically the guts wall, which are the structure of this part of the cell are represented here on your right. So basically this what happened when oil got together. Okay, so this part here can be resembled to the oil part of our body. we have two different mechanisms that our body uses to absorb drugs. One is called active transport. So the body consumes energy in terms of ATP, okay, to take all the drugs through the cell and dissolve in the bloodstream. And then we have a passive diffusion. Okay, so the molecule just freely pass through. It's like for us to pass through an open door. Okay, it's very easy. You don't have any interference. Meanwhile, in the active transport, you need to press the button to open the door and get through the cell. Okay. So, what is active transport? Active transport can be represented as someone take these very heavy. This is the drug. This is the transporter. So take these molecule through the cell wall. Okay. But what happens if you don't have this kind of transporter? MANAC apparently doesn't have any transporter. Glucose is very easily absorbed by the body because we have a sugar transporter, glucose transporter. So, but MANAC probably doesn't have this kind of transporter. So in order to get these drugs into the cell, we need a lot of drugs, okay? And these reflect in why, when you take MANAC, you have to take a high dose of this compound. You have to take a lot of dose, and usually with every drugs, high dosage means more problem, side effects. Okay, more the effect and the side effect of the drugs are highly dependent on the dosage. Okay. On the other hand, when you have a passive transport, you can try to think about the passive transport. You can push this barrel here, this stone here through this to the valley. It's very easy process. You just need a little push, and this part here goes down. And the same happened for lipophilic drugs that resemble oil, going through the cell. Okay, it doesn't require any effort from the body. When it's there, it just freely goes through the cell membrane. Okay, as represented here. I represented the drugs as an oil drop, and when you go, it goes easily through the cell wall. Okay, and basically you just resolve the problem of the gut absorption. And here the first rationale of our drugs, we take MANAC, okay, and we replace the OH group over here, responsible for the ability of the MANAC to be soluble in water, with this group here. So 1, 2, and 3, but especially this part here, make the MANAC more similar to an oil drop rather than a water molecule. Okay, so in this way, this molecule can easily cross by passive transportation the cell membrane in the guts as well as in the cellular target. So we have basically we think that this approach can solve the problem of having high dosage of this molecule in the patient, if this hypothesis will work. And this is the first part, the first point of the rationale of this approach, our approach on the prodrugs. The second is, here I just is a snapshot of the first part of the sialic acid synthesis that involved the GNE enzyme. This enzyme does essentially two things. It takes this compound here, N-acetylglucosamine, and basically change the position of this part in red from down to up. And then immediately when you have this part, this molecule here, it adds some phosphorus, is the pink moiety over here. Okay, so the same enzyme does two things. Okay, so which is our approach in this case? Here we have what happened now. You have your MANAC, okay, the molecule that's a clinical trial now. As I said before, it's a very polar molecule. Okay, so it likes to stay in water, and for this reason has a low permeability. Okay, that's why you need to have 3 to 6 grams twice a daily of this compound in order for this molecule to pass through the cell membrane, okay, in such a concentration that is therapeutically effective. So when you deliver this molecule here, you are at this point in the sialic acid pathway, okay? So in this case, you deliver this part of the molecule, this molecule here, which bypassed the first step of the GNE enzyme. But what happens if also this part here, the second part of the enzyme, the part that attacked the phosphorus on this molecule, is defective? This part here will be also defective. So the final product won't be produced in sufficient amount And as a consequence, the sialic acid won't be produced in sufficient amount. So what we thought in our case is to attack the phosphorus already on the MANAC. So we have a lipophilic molecule, okay, they easily cross the cell membrane. And once this blue part and green part will falling apart, we have the mechanism for these two part here, they just falling apart inside the cell. Basically, we delivering the final product of the defective gene enzyme. So with one molecule, we are basically
Dr. Fabrizio?
Yes.
Is it possible for you to wrap it up in another minute or so?
Absolutely. Absolutely.
You,
so no problem. So basically, we just deliver this by passing these two enzymes, these enzymes. Here, just the quick results. So basically, we test one compound, and basically we saw this compound was able to improve the concentration, the production of sialic acid in GNE myopathy cell model. Okay, I will skip this slide because just first I would like to give you, to conclude just my view about the as a scientist working with patients. I think, as Michela suggested, we really need the patient because are the people that really need the treatment, but also they know the disease much better than the scientists. And this can be a huge help, especially from our side, the very early stage in the drug discovery, to be able to design a proper drugs with the right properties and to design a proper drugs. So patient collaboration, just to summarize this slide, is very, very important. And we are really fortunate to have Michela and Jenny patient on our side in the development of this compound. Finally, just a slide to thank Dr. Stephen Thomas and Patrick Crutcher, because these two guys are the one who came to us with these contributed to our development of the early stage of this compound. And of course, thanks, Michela, and thanks to you for inviting me and give the opportunity to share this with you. And of course, I'm happy to take any question I have.
So thank you very much, Dr. Fabrizio, for this wonderful presentation. And so we are really lack of time, but we can take one question. What I can see put up by Neetu Singh. She asked that when the ProDRUG-GNE clinical trial would start, when will the ProDRUG come in the market? And a follow-up question she's asked is that what is the difference between alpha and beta MANAC?
Okay, the first one, the GNE project itself is still in infancy. So we were expected at the beginning to, after 3 years of the project, to be able to start to reach the clinical trial, to be able to have a molecule that has a potential to reach the clinical trial. But unfortunately, COVID-19 has been a big blow for this, and we are a little bit behind. But the idea is to have, after the 3 years, be able to, hopefully to start the clinical trial of this. For the market, honestly, we are still far away from this. And for the second question, honestly, alpha and beta MANAC, but alpha and beta, you mean the numeric position, I guess. Because in this case, I don't think there should be any problem in this. Because I think the GNE is able to process both the alpha and beta anomers of the glucosamine and therefore the mannose-amine that is produced. Is that answer your question?
I hope I'm sure the question has been answered. So we don't have time to take further more questions. Thank you so very much, Michela and Dr. Fabrizio, for this wonderful talk and insightful presentation.
Thank you very much.
We will directly head on to the next session.
Maybe, Rishabh, sorry to interrupt, maybe people can ask questions in the chat and maybe other speakers can answer in the chat.
Yeah. You can also contact me
I think that's great.
in private, it's fine.
Great, perfect. So we'll jump on to the next session. So the next session is consisting of 3 people. First one is Dr. Ram